Good morning, good morning, good morning. Hello, Zoology 2700 enthusiasts. Hello. Now, I am seeing... Hello. I would like to take a moment this morning, take a second and appreciate what has basically turned into kind of like festive socks. Well done, you. Today, we're going to be talking about a mystery, and it has to do with socks, as all good mysteries do. And so, we're going to get going. I have the chat open in the on my other screen, so we'll keep checking in to see if you've got any questions. And if you're viscerally connected to your socks here, take a look. I've screen grabbed them, and I'm going to clear, and we're going to get underway. Okay. So, let's clear all drawings. Here, we go on Friday, a snowy and cloudy Friday. We're going to be talking today about, we're going to jump into closer and closer to our parts of the Dunn phylogeny, of this evolutionary hypothesis that groups all animal lives. Our home is, of course, in one of the more boring parts here, represented by the whale. But we're entering the bilateria. So bilaterally symmetrical animals. We are leaving the radially or lacking symmetry groups, the early branching ctenophores, sponges, placozoans, and cnidarians. And we're going to spend this morning in the bilateria and then inside of this particular, but not diverse, but interesting and important group called the Xenocelamorpha. And if you say that as a Hermione, you can probably lift something. Xenocelamorpha. Wingardium Leviosa. So, before we get to the specifics of Xenocelamorpha and a lot of the stories that we have to talk to you today, I wanted to talk about the apomorphies that kind of combine our home in the bilateria. They are that we are, as the name suggests, bilaterally symmetrical, so that there's a mid-sagittal plane with left and right mirror images, and we have dorsal and ventral sides, so left and right, stomach and back, front and back. This is a characteristic of this group. As you'll see, it's been lost in several cases. Cephalization, so a neural network, a complex, complicated, and centralized neural network. We'll talk about advantages and disadvantages, primarily advantages to that strategy. They're triploblasts. We are triploblasts. Endoderm, ectoderm, which we've seen in other lineages, and now mesoderm. We have a through gut, so we have a mouth and an anus, and excretory organs. And we'll talk as we proceed through particular examples of this group about different ways of excreting waste and byproducts of metabolism. So nephridia, protonephridia, and metanephridia. And there are particular chapters. Today, I'd like you to think about, in particular, chapter 4, which you've already been through, but there's a lot of the base, kind of overarching, not even base, overarching themes that are captured there, and then chapter 9. Now remember, as you're thinking about the Dunn phylogeny, I'm going to encourage you to think about a simplified version here. And so we've been inside of these phyla so far, the ctenophores, the comb jellies, the sponges, the placozoans, the jellies and corals. And now we're in this large clade here, from this node up. And this is an exciting place to get, not least because it's the most diverse once we get into the arthropods, but because our ideas, as that quote says at the bottom, about the relationships within the bilateria have undergone major paradigm shifts within multiples. And the author used singular, I'm going to use multiple shifts in the last decade. One of them that we're going to talk about today is this group, so the Protostomia, which is a group that contains our clade, the deuterostomes here, so us, and the echinoderms, then the spiralians, so your mollusks and others, and the ecdysozoans, so your arthropods and others. And you'll notice in that grouping, you see how Bilateria hangs out here. What do we do with the Xenocelamorpha? Now, what do we do with the Xenocelamorpha is not a great idea or a great title for a mystery novel, so we're going to call it The Mystery of the Purple Sock. And you have no idea how much that delights me. And in fact, I was meant to be wearing these, but when I started teaching this course, I went out and bought purple socks. And because if we were sharing this as a lecture theater together, I would have my shoes off and I'd be wandering around in these socks, but we'll just keep them there so you can remember what we're talking about. And I love the music, so we're going to do it again. What is the mystery of the purple sock? Well, to tell you about the mystery of the purple sock, first of all, we have to share some knowledge about the Xenocelamorpha. This is a group of, as you can see, colorful, tiny bag-like organisms. This is actually Xenotrebella here, and these are a lot of examples of aceals. So we're going to talk about that. But what are some of the characteristics of the phylum that groups the aceals or the Acelamorphs and the Xenotrebellids? They're listed here, that they're soft body, they are dorsoventrally flattened, they're acoelomate, and also almost exclusively marine worm-like things. They have an epidermis with particular cell types that aren't found in any other metazoan phylum. They have an incomplete, I love that it's specified here. Look at point number 3. An incomplete gut, i.e. lacking an anus. So you can imagine that they also lack some other things. Discrete organs, no discrete circulatory system. They have protonephridia, no proto or nephridia or meta, or no organized gonads. Their cerebral ganglion is simplified. They have circular and longitudinal muscles, which means they can kind of inch their way around, but in the same way that basically the jellies did. They have, if you want to think about, this is kind of beyond, but if you're interested in developmental genes like Hox genes, they have them, but they have fewer of them than all of the other metazoans. And if you know anything about Hox genes, that's kind of amazing because they are deep things embedded across millions and millions of years of time, shared across multiple disparate, very distantly related organisms. And no larval forms. And let's go back. This is a video of Xenotrebella bacchi, filmed by Max Telford, who's an author that you're going to see. And look at that. This is, I don't know, you can tell me about it in the chat. I'll jump back over there in a second. But you remember we talked about the jellyfish Ephyra and strobilation, and we thought maybe that was a competitor for the cutest invertebrate that we're going to see. I'm not sure if you think this is a competitor or not, but this is actual scientific footage of Xenotrebella bacchi, until recently, one of the only species known of Xenotrebella. And in fact, a source of scientific confusion for nearly 100 years. So the Xenocelamorphs, you've seen this, the box capture from Brusca, but basically they have no gut, they have unique cilia, which is they have no discrete organs, no known larval forms, circular and longitudinal muscles. Very divergent groups, the Xenotrebella and the aceals. And most of the lecture we're going to talk about Xenotrebella. So I'm going to tell you a little bit about the aceals because I feel like we have to do them justice. And part of what we're here to do is to celebrate the diversity of form, function, and evolution. And so if I don't talk to you about the aceals, who will? Well, clearly Sherry will. But if neither Sherry nor I talk to you about this, who will? Somebody must. So take a look. This is a cross-section of one of them. And you can see that, just remember the statuses that we saw on Wednesday with ctenophores, another group that made me very excited. They had this statuses. So this is dealing with response and orientation to gravity. So simplified, longitudinal and circular muscles, so they're kind of pulsating around, but they are responding to different environmental stimuli, classic and large ones like gravity. Here, you're taking a look at what does a kind of a creeping around Xenotrebella look like? Well, there's a video from a remotely operated vehicle off of the coast of California looking at one that's on the bottom, and then you've got some CT tomography that's looking, spinning Xenotrebella around, just in case you thought it wasn't interesting when you watched it move. Here you can see those are the features: frontal pores and a mouth and no anus. What you gonna do? Xenotrebella has had a checkered history of placement throughout time. This is maybe a theme that you're coming up against as we walk together through the phylogeny, the tree of animal life. And so Turbellaria, and we'll talk about Turbellaria when we get to the platyhelminths on next Monday, or next week on Monday. Xenotrebella used to be considered to be a flatworm. Turbellarians, which is still a word sometimes that we use to describe how you move, again a theme that you've seen throughout the course, even dating back to the protozoans, it's a paraphyletic group. So the aceals and the Xenotrebellans used to be grouped in here. And you can see, even according to this older phylogeny that was captured from Rupert, Fox, and Barnes, that word doesn't include all of the things. Aceals, mouth, no anus, these lateral branches associated with increasing surface area of the, where are we here? Phagocytic gland cells. They have a small gastrovascular system, so slightly different than, and more diverse than the Xenotrebellids, potentially a little bit more complicated. Now, that's a bunch so far, just of like, here's what they look like, not much. Here's what they do. And now I'm going to jump back over and see, is Xenotrebella different from Xenocelamorpha? Xenocelamorpha is the phylum that contains Xenotrebella and the complicated and myriad history of Xenotrebella. This tiny little creeping worm was discovered in 1915 by a Swede named Sexton Bach off of western Sweden, right here, at about 50 to 200 meters in depth. It was described relatively quickly. Some of you are finding in the naming assignment that the time in between the discovery of the sample and the confusion about what species is what, that happens at time X. It often needs years, decades, sometimes many decades. The intervening time exists between when the species is actually described. Now that description, amazingly, of the Xenotrebella, of this, was 29 pages long. Now, I don't read or write Swedish, so I don't know if that's just a characteristic of how complicated the language is, but I don't know that if you paid me, I could come up with 29 pages about that particular organism. But they did. So Xeno, in this case, as somebody mentioned this, as you said, alien, Xeno means strange. And so you're right. The idea with this seemed to be a worm that was a strange thing. Now, only 1 to 3 centimeters long. It's ciliated. It seems to be a diploblast, so not a triploblast, and no anus or distinct organs. Now, Box and Westblad, the discoverer and the describer, the authority, so well-timed question in the chat, were Platyhelminthes taxonomists. They were flatworm taxonomists. And so they called it a platyhelmuthworm. And I think this is a little bit of what's called colloquially the law of the instrument. If you're trained to work with hammers, you see nails. So in this case, now granted, this is very much looking like a platyhelmuthworm, but they were trained as platyhelminthworms, and that is what they saw. That's what they described. Now, so since then, Sweden has been the only place we've seen it. This is a video and a description of this is how you collect Xenotrebella historically. You have a dredge or basically, so this large canister here is lowered off the ship, lowered down to the bottom, and dragged across the substrate. Then it's retrieved, this box, this Kleenex box that's now full of kind of the top layer of substrate off of the bottom of the ocean. And then researchers filter it with a hose onto a bucket, and then they sit on the deck of the ship and they sort through and they make little piles and say, here's where all the cnidarians are going to go, here's where all the ctenophores, here's our sponge pile, here's our fish pile. And then people make smaller. So they take a fish pile and make it into smaller particular families. And you can imagine that if you're a soft-bodied dorsal ventrally flattened or radially symmetrical like a cnidarian or a ctenophore. This process of collection kind of tends to homogenize you, tends to alter you and make you basically some of the sludge that maybe is left over here after being washed off. So easy to overlook, hard to understand their biology when what we see basically is what they look like when they're caught on a sieve. So that dredge brings up the mud, and this animal's, remember, squishy, only 1 to 3 centimeters long. Now, I'm going to ask you a question next in a Zoom poll. Now, I'm going to back up for a second. This is how we've seen them to date. Just like we talked about ctenophores, they're hard to preserve and they're hard to get DNA from because they're soft-bodied and because of historically we preserve things in formalin, which is very bad for the preservation of DNA. 1997 was an interesting year for a bunch of reasons. For me, let's see what happened in well, I think my hair was lower than here. It was big, glorious, curly red hair. And the very first genetic sequences were published from a collection of Xenotrebella, and it was published in Nature. People were excited about this tiny ciliated white worm from the coast of Sweden. And I'm going to ask you in a Zoom poll, based on this phylogeny, so you can see here, this is where the sequence came up. Now, based on that sequence, based on the placement of that sequence in this phylogeny, what does that tell you? What did that sequence tell people about what is a Xenotrebella? So the poll is open. You've got some names bouncing around the top there. Feel free to Google them. So your suggestions are, is it a cnidarian? Is it a jelly? Is it an echinoderm? Is it a chordate? Is it a Platyhelmuthworm? Is it a nemertean worm? Is it an annelid? Is it a mollusk or is it a sponge? Now remember, so that's a very simplified phylogeny. This is what phylogenies look like in Nature in 1997. And the red line is where the Xenotrebella sequence popped up. I was just spit on. My water bottle is speaking moistly. Keep 2 meters apart when you're speaking moistly. All right, I see only almost 50%, now more than 50% of you have voted. Salut, petit monstre. Let's see if we can get up to 75. Awesome. Let's take a look at those results. And the category that the majority of you thought is actually, that was the conclusion that they made in the paper. These DNA sequences say Xenotrebella is a bilaterian. It's not a deuterostome. It's not sister to the Nephrozoa or like the earliest branching bilaterian. This is a mollusk. Now I know we haven't been to mollusks yet, mollusk land. We haven't talked about mollusks, but I would ask you to consider what you know about mollusks and say, this is a surprising result. For one, no shell. So these 2 articles in 1997 in Nature used some ribosomal and mitochondrial DNA sequences to propose their hypothesis that Xenotrebella was a mollusk. And so one of them did the DNA work and the other reported that there were mollusk-like eggs, that they found and discovered in Xenotrebella based on these kinds of dredges off the shores of Sweden. So this is a quote, so that Xenotrebella bacchi is neither a sister group of Bilateria, nor from any other, they say basal, we don't like that word, basal metazoan group, but it is a mollusk related to or within the Protobranchia, and that's saying the particular lineage. It jumped in there closest to the bivalves, so Protobranchia. Now, we're not done, because 6 years later, people examined this was a confusing thing. And so people continued to collect Xenotrebella, and they found, using 3 genes that Borlat et al. Did 6 years later, that the samples that had been published in 1997 weren't actually DNA derived from the Xenotrebella. That was DNA from what the Xenotrebella had eaten. And so those sequences were contaminants or exogenous DNA based on what the Xenotrebella had consumed. And their analysis then moved the ball out of to say, so Xenotrebella, where does it sit? First hypothesis is a Platyhelmuthworm. That's an older hypothesis that predates the use of DNA. Second hypothesis is that it's a mollusk. The third hypothesis, now that Sarah Borlat and colleagues published, remember Max Telford, who shot the video? There he is, Maximilian J. Telford. That's a splendid name. Is the hypothesis that it is not a Platyhelmuthworm, not a mollusk, that it's a deuterostome, like in our lineage. So related to hemichordates and echinoderms. And there is a look at the gene and the small number of still a small number of genes, 3, that they looked at its placement and said, well, here, it's not a chordate, it's kind of sister to hemichordates, and there's the Lophotrochozoa, a name that would contain mollusks, and the ecdysozoans. So it doesn't fall, it's not an early branching thing here, according to this phylogenetic hypothesis. So we've seen a huge kind of Andy Murray style tennis match as the placement of this tiny 1.5 ciliated, 1 to 3 centimeter long ciliated worm has bounced around the animal phylogeny. And this is an editorial that Henry G., who was a longtime editor at Nature, wrote about the fact that you aren't what you eat. As we reflect on Xenotrebella's newly exalted state as a deuterostome, it is worth recalling similar examples of taxonomic misclassification, or what a meme-based world would call taxonomy fail, #. Aristotle classified tunicates as mollusks, and the Amphioxus was first described as a kind of slug. There is no shame in having once been classed as a mollusk. Many of the best deuterostomes started out that way. I like his writing, that's why it's there. So let me jump back over the chat for a second. Exactly. 29 pages about Xenotrebella, but you're right, Sherry. Darwin wrote 329 pages about vegetable mold and earthworms. Oh, and then someone who knows about Swedish is not that difficult to write. So 29 pages were probably 29 solid pages about the Xenotrebella. Yowza. Looks like a cashew. I like that. I hope those socks are clean. Well, this is Zoom land, right? We presume the socks hope that Smith is clean. Who knows? All right. Good. Okay. Jumping back over, and we're going to keep going. So Xenotrebella starts out as a platyhelminth flatworm. Then we think it's a mollusk. Then we think it's a deuterostome. And there's an editorial written in Nature that says, look at that. Lots of great things started out as mollusks and ended up as deuterostomes. But wait, so is that the final position? If you've read through, you remember that shout out I did earlier to the Dunn et al. Paper. It's not a completely understood position as of yet, or as of 5 years ago. So these are the two competing hypotheses. We have that the Xenocelamorphs are a sister group, the earliest branching bilaterian. Okay, so sister to the Nephrozoa. You can see there is the red ball indicating Nephrozoa that links chordata and deuterostomes and protostomes. So, and there's some waxing lyrical terms that Haynal and all have used to describe the odyssey through the tree of life. Or there's Philippe and others, as of Nature in 2003, I suppose, with the 3 genes that came up with the hypothesis that Xenocelamorphs are actually deuterostomes. So I'm going to ask you to use your Zoom annotate tools here in a second. So bilaterians have a nerve cord. We know that Xenocelamorphs don't. Bilaterians have a nerve cord. So use your annotate tools, where would the nerve cord have evolved according to this hypothesis? So use your Zoom annotate tool. Okay. I see. Okay, so that's great. Let me open my annotate tool so that I can save that. Two hypotheses here. One of them is that they appeared, and I'm going to clear that. No, I'm not going to clear, but just stop annotating for 2 seconds. There seems to be two hypotheses. One of them is that it occurred here with the Nephrozoa, or one of them is that it occurred with the deuterostomes. Now remember, the bilateria is not just us, right? It is, so protostomes include arthropods and mites and onychophorans and tardigrades. So according to this hypothesis, it's actually here, and this will appear, but it is right here. So the idea was that it evolved once Xenocelamorphs branched off. Then we see the evolution of this of a nerve cord and cephalization. Now, clear all drawings. I'm going to ask you according to the next hypothesis. So this is the alternate hypothesis. This is Felipe's hypothesis. According to this, where did the nerve cord evolve? Yeah, that's it. That's right. Well done. Let me save that also. Super. Clear all drawings, and let's keep going. There's just a page for you to have later on. So according to the first, it's there. And according to the second, it's there and then lost here. And that's what the X inside of the yellow ball means. So the consequences of our understanding of neural evolution, which we talked about already with the ctenophores, and perhaps the ctenophores being an example of the independent evolution of a nervous system. Here, our understanding of a cephalized, complicated nervous system with a nerve cord is greatly affected by where we put this little 1 to 3 centimeter long flatworm. So, according to this hypothesis, excretory organs, the top one, as sister, and nerve cords evolved after the Xenocelamorphs separate from the Nephrozoa. So a little bit, I'm going to jump to the chat real quickly. I want to same thing else there. I want to just make mention that those kinds of questions, consequences, true, false questions, those are the kinds of things, as we've talked about, that the lecture quiz will be full of. So if there's a phylogeny, I would never expect you to memorize the phylogeny, but I'll show you it, and then I'll map traits on or competing phylogenetic hypotheses, and maybe I'll then ask you 2 or 3 questions about that particular arrangement of phylogenetic hypotheses or the consequences of that arrangement. Cool. And this, of course, speaking about the lecture quiz, this is the last lecture that will be included in lecture 1. The remainder are going to be included far down the line in lecture quiz 2. And the lecture quiz, of course, is open on Friday next, the week today. Now, we're not done with the story and the mystery of the purple sock. We haven't even come to the part of the purple sock part yet. These are 2 papers. Remember, 2 papers in Nature in 1997 sent us down a mollusk trail. In 2016 rather, in February, while we were teaching this class. So when I taught about Xenocelamorphs, I was vibrating with excitement talking about this, because we were talking about research that had come out like earlier that week that had been published. 2 things came out. One of them was talking about the phylogenetic placement of Xenocelamorph, and one of them was describing the description of new species of Xenotrebellans, both of which are important, I think, to you today. So 2 things, videos and pictures here. One of the things was that these were they're a global set of researchers, but the Xenotrebella were sampled this time not with dredges, but remotely operated vehicles. So gently, as you just saw, that little proboscis set off of the front, controlled by a ship on the surface, and then used to gently aspirate. This is a Xenotrebella that it's aspirating. So this is essentially a discovery moment of a new species as it's collected, but it's not going to be homogenized in the dredge. It's collected gently, placed in a single container, so the likelihoods of damage or contamination are super reduced. It also means that because we have these classic remotely operated vehicles, we have videos and we have pictures, and we understand more now about their biology. So one of them, the new elements in these studies in 2016, this is the first time. Remember, Sweden was the first time they were found. This is off the coast of California near Monterey Bay. They were also very big. So 3 new species were described. The first time in the Pacific Ocean. Some of them were very big. Some as long, almost as a ruler. I said 3. Sorry, all 4 species lived on the seafloor. Some were very, very deep, almost 4 kilometers deep, and required remotely operated vehicles equipped with a slurp gun. That is a slurp gun right there. And they were able to extract DNA out of all of them, and they identified kind of multiple clades or groups, shallow and deep, associated with where they were found. And you can see here, so these are the 2 clades. So Baki, this is the Swedish one. One of the new species that they discovered is Zalandorum, Halandorum. Over here on the left, that's shallow, so less than 650 meters. And then in the deeper parts, up to like a mile or almost 4 kilometers deep under the ocean, there are the monstrous, the profund, and the churro. And so 2 deep lineages. What does that suggest to me as an evolutionary biologist about the diversity of Xenotrebella around the world? Well, for one, it suggests to me that there's a lot of unknown diversity that we have yet to run into. Now, some of you have finished your species naming assignment already, and some of you are still fighting with it. And I hope that that fight is going well, because the intent with it was that it was relatively quick to do. I'm going to show you, because you've been thinking about species description. This is the species description for Xenotrebella churro. You can see the etymology here. That this is the picture from the remotely operated vehicle before they slurped it up in the slurp cannon. Why was it named churro? It was named churro because it resembles fried dough pastry called a churro. It has 1 holotype, so 1 sample has been collected. There's no mention of any other paratypes in this description. The sample's female, specific lat long and depths from where it came, on it collected and via slurp, so that they know something about the quality of it, and the dive, the date, and the ship, and then a description, and the fact that they've also used other bits of DNA, exogenous DNA from a co-occurring bivalve present in the tissue. So this is biological information embedded in that species description that suggests that whatever else Xenotrebellas are doing, whether they're living shallow or deep, they like eating bivalve mollusks. So this suggests, and that the 2 paired, so the Canada paper and the Rouse paper, so one's a species description and one is the phylogenetic position. These are the results of the phylogenetic position study, that it supported the view that, as the title of the paper, Xenocelamorpha is the sister group to Nephrozoa. They can't be accused of burying the lead. That's the results of the paper as well as the title. So outside of the bilaterian, we have Cnidarian and other lacking bilateral symmetry lineages, earliest branching Xenocelamorphs. And so it suggests these things about the evolution of some complicated traits that we see within animals, that we see that they have a guts and a nerve net that is much more like the Cnidarians, but they do have a mesoderm, and they are bilaterally symmetrical, and they lack nephridia. So the retention of some of the derived traits that characterize the group, or the possession rather of some of the derived traits, retention potentially of traits that are more similar to earlier branching lineages, or an alternative hypothesis would be the convergent evolution towards those characteristics of an earlier branching lineage. So that's this category here. Now, as you might imagine, this was big news. Maybe you don't imagine it, but this was big news. This is the BBC. The mystery of the purple sock has been solved. Boom, boom, boom. And we talked about this not just in the Beeb, but we talked about it within the CBC. So this is As It Happens, which is the radio program that I will always have time for in my life, and is currently hosted, and I hope forever hosted, by Carol Off, tweeting about a text, this paper, this study, these 2 studies, and making a taxonomic fail. Stuff of nightmares. I disagree. A deep sea mollusk with just 1 hole looks like a purple sock. So they're clinging in on the fact that there's no anus, but they've called it a mollusk. It is purple, but it is not a mollusk. So what was I to do? Sit quietly? So I tweeted back at As It Happens and said, not a mollusk. It's a teachable moment. They responded. Thanks, Alex. Of course, we'll fix. Then they fixed that it deep sea creature. So with just one hole, that looks like a perfect song. And that was also the start of a bunch of different CBC-based discoveries on As It Happens. But they, yeah, if you don't know As It Happens, you check it out. 6:30, CBC Radio 1. So we talked about butts with Tina Belcher and Tina Forrest the other day, but these deep sea worms, this is, again, popular press that's seizing in on the fact that these are the earliest branching bilaterian that doesn't have an anus. So this is part of our family, but it's a very divergent, early divergent. And then what can it teach us about characteristics of this group that we call home? Yes, people literally wanting to eat churros now. Yes, please. That would be lovely. Share. All right, jumping back over. We're not done. We will be done shortly. But in 2018 and 2019, the discoveries using remotely operated vehicles that allow the retention and collection and sensitive collection both of deep sea organisms that otherwise would have been homogenized or damaged in dredges, and the increasing frequency with which ROVs are used and slurp cannon technology are used allows us to video and collect things. And so we're finding them. This idea that we had for many, many, many years, right back to Bach's first description was, or sorry, not Bach's description, because it's called Bacchi, right? His first collection in the 1800s and then description in 1949 was that this seemed to be a lineage in marine Arctic waters. So we've now seen them in the Western Pacific by Monterey, California. We now see them off of the coast of Japan, and we're finding, not remember the exogenous DNA, so that we're pretty convinced that they eat mollusks for a couple of different reasons, a couple of different studies. As we can collect them more sensitively with these slurp cannons, we're discovering more and more about other species that live on them. And so this other study here on the right-hand side, I'll tell you about in a second, where a representative of another understudied phylum was found parasitizing one of the Xenotrebella. So this is a new species of Xenotrebella that was discovered off of the Japanese coast. And you can see this is where I took some of these scanning, the tomography. And you can see it is as gorgeous, let's say, as all of the other ones. But I want to make a note that these both actually where Xenotrebella bacchi was originally captured in Sweden and where this one was found and discovered in Japan, and where the churro and others were discovered off the coast of Costa Rica, these are all within an hour of a marine station. So we're still dealing with, this is an analogous to a terrestrial biologist. This is me still describing new species that I can find just by parking my car at the edge of the road, that you're not exploring the deeps and unexplored aspects yet. So how many more species of Xenotrebella yet remain to be described? Many, many more. Now, a word about the Nakano et al. Paper on the the right-hand side here. This is a new species of Orthonectida that parasitizes. This is where the Xenocelamorphs are here. And orthonectids are a group of spiralians that we actually spend very little time with. They're parasitic, they're not diverse, and this is what they look like as they parasitize Xenotrebella. So a minor phylum of microscopic marine parasites that parasitize aceals, Platyhelmuthworms, soft things that live in the ocean, and it's present in the host animal, and in this multinucleate plasmodia, and it's a very simple morphological system. So discoveries based, like stacked on discoveries, and we're not done. So this is a video from another expedition. The first one that we talked about with churro, that was a Mabari expedition. This is one from the Schmidt. This is Greg Rouse, who was actually the author, the Australian scientist who's one of the species authors, describing this now to a if you have a bad internet connection, you might think this is Smith continuing to show us Xenotrebella churro or profunda or some of the other ones off the coast of Mabare. This is not. These are captures of Xenotrebellids off in a new location off the coast of Costa Rica. And a lot of these.
That's a Xenotrebella. Oh, wait. Hold, stay there.
This is the
Okay, we'll probably get one of those.
that last voice is a colleague.
Yeah, let's get video of this. Greg will want video.
Yeah.
And this is the actual video. That was the actual video of them discovering it. So you can go and
Holy shit.
Sorry. Is that loud?
Holy shit.
That's Greg Rouse coming to the bridge and being shown the Xenotrebella that they've just discovered. That's you just watch, and it's captured there because these trawls are streamed live often on the interpipes. That's him responding, as many scientists do at the moment of discovery. Holy shit, this is a new species. This is going to be a Xenotrebella. And this was also discovered while we were teaching this class a couple of years ago on a dive off the coast of Costa Rica here. So for the longest time, we thought they were restricted to Sweden, but now we see them kind of distributed around the world in each case, and each one of, except for Costa Rica, actually. Costa Rica is the first one that breaks the within an hour or so of a marine station. There is so much to discover within this group, and consequences towards our understanding of neural evolution, of the evolution of a through gut. Let me jump back over here. Amazing. Now, that's the story of the Xenotrebella. That is the solving of the mystery of the purple sock. And what I'm going to do is say, have a great week. And I hope that you do things for yourself. Do something physical, do something that's not your schoolwork, not your job. Do something to take care of yourself, do something to take care of somebody else around you when you're, once you've done that. And maybe go and listen to the Arctic monkeys' knee-high socks in honor of the Xenotrebellids and the mystery of the purple sock. And what I'm gonna do is I won't turn off the Zoom link. I'm just gonna turn off the video and the audio, stop sharing the screen, and then I'll come back in 10 minutes or so. And if there's questions, that's the start of the lab hours. So I'll see you Monday, and I'll make things kind of asynchronously available in the background, and see you soon.